All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. Next slide, please. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support make this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10 to receive a 10% on your membership sign up. ASPB members get access to all this webinar and all the amazing benefits that ASPB provide. You can learn more about ASPB and the opportunities that we offer our community at aspb.org. Next slide, please. In addition, we have many other webinars that we've recorded. So if you're not already following the Plantae YouTube channel, be sure to do so. And if you're looking for additional great plant science content, whether it be focused on career education or many other exciting talks, be sure to check it out. Again, this webinar is recorded and you will be able to watch it afterwards and share it with your colleagues. Next slide, please. As you may have heard, registration for the Plant Biology Conference that the American Society of Plant Biologists hosts is open. This year it's taking place in Milwaukee, Wisconsin from July 26th to the 30th. And so if you haven't registered or are interested in learning more, you can do that by going to plantbiology.aspb.org. It is a very unique event. It allows you the opportunity to connect with people from all over the plant science community, where you can obtain knowledge, network, and then learn all about the exciting things and advancement that are happening within this plant science space. It's also an opportunity that is open to all career levels. Next slide, please. Before we get started, I'd like to go over a few housekeeping items to make sure that you have you get the most out of your attendance in today's webinar. If you are experiencing technical difficulties, you can use the Q&A box by putting that in, or you can email me directly at jpadilla@aspb.org. If you have a question for our guest speakers, please add them in the Q&A box also. Our moderators will be sharing them with our presenter during our Q&A session. If you have trouble connecting or need to leave the webinar early, as I mentioned, this webinar is recorded and you can watch it later on our YouTube channel. So again, be sure to follow our Plantae YouTube channel to receive notification when new content are posted. And so without further ado, I am now going to turn it over to our Plantae fellows who will be facilitating today's sessions. With that said, take it away, Plantae fellows.
All right, hello and welcome everybody to today's Plantae webinar entitled The Role of Plant Scientists in Space Agriculture, Gardening the Galaxy. We're so excited to have everybody join us today from around the world, and we hope you learn a lot from our incredible lineup of speakers. So as we look towards long duration space missions and the possibility of one day reaching other planets, one thing really becomes clear: humans can't go far without plants. From providing vital food and oxygen to recycling nutrients and stabilizing life support, plants are essential to sustaining life here on Earth and will be essential companions in the future. But growing plants in space isn't as simple as packing some seeds and potting soil. Challenges like microgravity, radiation, and the availability of nutrients, light, and water are conditions that will subvert what we really know about plant biology here on Earth. And that's where plant scientists will come in. The field of space agriculture is no longer the stuff of science fiction. It's an interdisciplinary study at the intersection of plant science, engineering, biotechnology, and astrobiology. By understanding how plants respond to conditions off of our planet and how we can adapt systems to support them, we're not only preparing for life in space, we can learn how to grow more resilient, efficient crops here on Earth as well. So in this webinar, we'll get a chance to hear from each of our speakers, and then we will open the floor to you, the audience, for a question and answer session. We will first hear from Dr. Simon Gilroy, a professor in the Department of Botany at the University of Wisconsin-Madison. After that will be Dr. Jenny Mortimer, a professor of plant synthetic biology in the School of Agriculture, Food, and Wine at the University of Adelaide in Australia. And our final presenter will be Dr. Veronica D'Amico, a professor of environmental and applied botany at the University of Naples Federico II. This webinar was organized by a team from the 2025 class of Plantae Fellows. If you could go to the next slide. We have Marvin Jin, a PhD student in the Research School of Biology at the Australian National University. Ved Prakash, a postdoctoral fellow at the Ohio State University. Abira Sahu, a scientist in the Department of Environmental Health Sciences at the University of Alabama, Birmingham. And myself, Maya Sealander, a master's student in the Division of Biological Sciences at the University of Missouri. Next slide, please. I have the pleasure of introducing our first speaker, Dr. Simon Gilroy. Dr. Gilroy is a professor in the Department of Botany at the University of Wisconsin-Madison. His research team focuses on dissecting the molecular underpinnings of plant stress response signaling systems with an emphasis on the effects of spaceflight. He has completed 5 spaceflight experiments to the International Space Station and currently has active research programs on plant microbe interactions during spaceflight, the effects of space radiation on plant microbe interactions, and on how lunar regolith might be bioremediated to support plant growth for long-term habitation on the moon. Take it away, Dr. Gilroy.
Mm-hmm. All right. Well, welcome everyone. What I thought I would do, as we don't have a huge amount of time, is rather than spend a lot of time delving into data and responses, kind of give a little bit of an overview of plant biology programs in space and why there is a lot of plant biology in space flight research at the moment, and it's going to be true for into the future. So a lot of the in space research at the moment occurs here. There's this International Space Station, and there is a tremendous amount of plant biology which is going on there. A lot of model systems, things like Arabidopsis, and a lot of crop plants, leafy greens, tomato plants, peppers, and the plant biology has a sort of an outsized impact on space research programs because people have realized that plants are going to be integral to how we're going to be in space. There's 2 components to what's going on there. There's using the space environment for basic research to understand what it means to be a plant and how plants operate. And then there is the other part of it, which is using plants in space for some element of life support. If you're going to feed people, produce oxygen, clean the water, all the good stuff that plant services on Earth are providing us with, those we want to take into space because we really don't know any other long-term life support system other than what we have on Earth. Those 2 things, basic research and the sort of more applied end of things, the life support, things like biomanufacturing in space, those are all the same thing, right? You can't go into this really alien environment of space and get plants to perform and do what we want them to do without the basic research to understand what's going on. And the justification for the basic research is in addition to just generation of knowledge, we're going to be able to develop a plant biology realm in space, which is going to support long-term human habitation in space. So it's all part of the same stuff. But I want to spend a little bit of time talking about the sort of the basic research end of things, because I think we're going to hear talks about the bioregenerative life support, biomanufacturing, other aspects of space in some the other 2 speakers. So let's take plants into space. And the obvious thing that you can then study when you go into space is gravitropism. right? You go into the weightless environment of space, you take away the directional cue of gravity, and you can do the classic experiment, ask, what does gravity give to plants? I'm going to take that gravitational cue away and find out what is different for plant biology. And that is a great set of experimentation, and people have been working on using the space environment to understand that fundamental part of plant growth. And it's the obvious knee-jerk one that you can think of about going into space. But there's a tremendous amount of other stuff that changes when you take biology that has evolved on the surface of the Earth. It's always had that terrestrial environment as its entire evolutionary history, and move it into a truly alien environment. And so we have the effects of altered gravity, or we have the effect, what we would call microgravity, changes in the level of gravity. We increase the radiation exposure. We're moving away from the constant of the Earth's magnetic field. There's a lot of things that are going to change when we go into space. And so all of these fundamental questions about what plants are doing in those environments allow you to understand how those environments affect plants on the surface of the Earth, but are also absolutely fundamental to understanding how we're going to have a long-term plant presence in space. I'm going to just do a very brief introduction to radiation exposure, because radiation is a biggie. It's a really, really major unknown for long-term habitation of space. And I know that Veronica's going to talk a little bit about this later on, but just to get people thinking about that, this is a really big unknown. The ISS daily dosage, if you're an astronaut, is about 0.5 millisieverts, which is about 5 chest X-rays worth every day. Which it sounds like a lot, and it is a lot. It's not like melt your biology kinds of level of radiation. It's long, constant, elevated exposure. And it's a big unknown. And the one that we're really worried about is ionizing radiation. And just to give you radiation, before I started to work in this area, felt like magic to me. You know, radiation's bad, but so the ionizing radiation of space, once you know what it is, it's really easy to understand why we should be interested and worried about it. Ionizing radiation, let's take a star exploding somewhere in the universe and take a hydrogen atom sitting inside that star, and it's exploded. There is so much energy that that atom is ejected and the electron is ripped off. And so what we're left with is a proton flying through space at the speed of an exploding star. That is an atomic scale bullet, right? And if it hits stuff, it's going to do damage. And it's got so much energy that it'll hit stuff and carry on going, but it will damage what it hits. That's what the ionizing radiation of space is. Let's say that bullet, that atomic bullet, hits DNA, right? That would be the classic one we'd all think about. Then we would have a mutation. That's a direct effect of the energy from that proton damaging a biological molecule. It doesn't have to be DNA. It could be a protein, could be a lipid, but the easiest one that we always think about is damaging DNA, and we'll have a mutation. That was what we would call the direct effect of radiation, and it's important. But there is another much bigger effect of radiation, which is, if you think about it, most of the biology is water. So the thing that proton or that nucleus of whatever atom it is most likely to hit is going to be water. It will break apart the water molecule, produce free radicals, and those free radicals will do uncontrolled chemistry inside the cell. And it turns out that radiolysis of water is where most of the damage is going to come from, because most of the stuff that's hit is water. It produces free radicals, and it's a largely oxidative stress then within the cell, causing uncontrolled damage within the cell. It's all bad, right? Oxidative stress, many of you will already be working on oxidative stress in your regular ground-based research, right? It's part of how biology responds to stressful environments. It's just in this case, we've got this other source of stress, which is ionizing radiation. Why is it such a big deal? Well, it's because biology doesn't really deal with it on the Earth. So you are here at the moment on the Earth. The Earth has a magnetic field. Those nuclei generally are charged. When they're flying through space, they might be coming from the sun. They might be coming from an explosion somewhere in the universe. but they are deflected around the Earth by its magnetic field. So we avoid them. All right. We're going to now move outside that protective sphere of the Earth's magnetic field, and the exposure is going to go up. And we don't know what that's going to do to biology, because biology has never really experienced that. It's going to affect astronauts. It's going to affect plants. How do we get a handle on that? One way is to go into space, right, and do experiments on radiation in space. The other way is to mimic that kind of radiation on Earth. And that turns out that's very hard to do. But just to give you a feeling that a lot of space research is not going into space. It's working out how to mimic space on the ground. This is a postdoc in the lab, Buju Alptekin. She's at Brookhaven Space Radiation Laboratory. This is like one of those huge particle accelerators like at CERN. And the picture on the right is Buju with a bunch of Arabidopsis plates growing on plants growing on plates stuck as a target for the beam to do radiation experiments on the ground. Because going into space is a rare opportunity. So working out how to have analogs on the ground becomes a really important part of it. So remember Buju's name. She'll come back in just a couple of minutes. But you can do experiments in space, right? And so altered gravity is one of the things that we think about a lot for space flight. This is an experiment. Again, this is Arabidopsis basic research. This is a plant growth facility called the Veggie. It's on the International Space Station. The hand you can see on the right is the hand of an astronaut called Scott Tingle, and Scott Tingle is doing one of our experiments with our Arabidopsis in there. All right, but you don't get to do the spaceflight experiment without the ground-based research to get you to the point of having a good question and experiment to do in space. All right, and this is the conundrum that we're super interested in about spaceflight. This is on the International Space Station. This is Chris Hadfield, astronaut. He's just going to wring out a towel that is saturated with water. And on Earth, he would wring it out. The water would be drawn down by gravity and his feet would get wet. But if you watch what happens in space, water just moves and behaves in different ways than it does on Earth. So he's wringing out the towel, but the water isn't falling down because there's no gravity to pull it down. The thing which is dominating the movement of water there is the molecular forces of water molecules. It's hydrogen bonding, it's surface tension. If you look at his hand, you'll see the water has crept over his hand by capillary action, and the hydrogen bonding is sticking things together. So he's going to try and let go of the towel, and it's going to stick to his hands because the water, I always say in space, water is sticky and creepy. All right? It'd be just different for how it is on Earth. The other thing which is missing in space is there's no convection, there's no buoyancy. So this is what it looks like to boil a kettle on the Earth, left-hand side, and in space, right-hand side. All right? And we're going to make bubbles of gas within the water as it boils. But in space, those less dense bubbles don't rise. There's no buoyancy. All right? So water is going to stick to biology in space. And we know that as a phenomenon. So what does that mean for plants? Well, here's some work by postdoc in the lab, Arko Bakshi, and his ground-based research is on how plants deal with being flooded, with water covering them on Earth. And from a lot of bioinformatics and a huge amount of ground-based research, we worked out that a mutant in a calcium pump called CAX2 is resistant to flooding on Earth. We know that this sticky water flooding response is part and parcel of being in space. And so you can take a very extensive ground-based research program and move it into flight. And so we were able to grow the CAX2 mutant in space. The movie at the bottom is in space. You don't get to do your experiment. This is the astronaut Scott Tingle doing our experiment for us, right? Photographing stuff and harvesting plants. We get the plants back and do things like exactly as you would expect for a normal groundy sort of experiment: image analysis, RNA-seq, glycomic profiling, to show that things like cell walls and defense and oxidative stress responses are altered in space. Right? So you have to have, there's a very deep ground research that feeds into the space flight research. And the last thought I'd like to leave you with is that a lot of you can if you're not into the field, you can kind of get this feeling that space research is very niche, very focused. And if you get into it, you are setting yourself up only for a career perhaps with a space agency or somewhere like that, right? Because kind of weird biology. But Arko Bakshi, spaceflight researcher, starting his assistant professorship in Wake Forest in January. Buju Alptekin does the radiation stuff, starting her assistant professorship at Ohio University in January. And I didn't get to talk about another postdoc in the lab, Sabrina Chin, who's working on lunar regolith and how to grow plants on the moon, starting her assistant professorship at the University of Tennessee in just a couple of months' time. So it's good science from space opens up a lot of careers, and it's not just with the space agencies. But that seems like a great place to stop and hand over to the next speaker.
Thank you very much, Dr. Gilroy. That was really interesting. It's my pleasure to introduce the next speaker, Dr. Jenny Mortimer. She is a professor of plant synthetic biology in the School of Agriculture, Food and Wine at the University of Adelaide in Australia. She's also the deputy director of the Waite Research Institute. She's chief investigator and University of Adelaide node leader of the ARC Center of Excellence Plants for Space. She's also an affiliate staff scientist at Lawrence Berkeley National Lab in the US and a director of plant systems biology at the Joint Bioenergy Institute in the US. After completing her PhD at Cambridge University in the UK, she began exploring how engineering the plant cell wall could deliver sustainable and economically viable biofuels. First as a postdoc in Cambridge and then as a research fellow at RIKEN in Japan, before joining the Berkeley Lab in 2014 and Adelaide in 2021. Her team's research focuses on understanding and manipulating plant cell metabolism with a focus on complex glycosylation. The goal is to develop knowledge and crops which contribute to a sustainable and renewable bioeconomy. With that, take it away, Dr. Mortimer.
Thank you very much. I am going to share my screen. Hopefully that's all clear in terms of slides. Okay. So my title today was thinking about how plant synthetic biology can enable remote-controlled environmental agriculture and biomanufacturing wherever you happen to find yourself. And that can be space, but it could also be a remote corner of, for example, Australia. But I think following on from Simon's last point about where space science can take you and how you can end up there, I'm pretty new to the area, so I've only really been working in thinking about space biology for the last 4 or 5 years. And my background really has been in other areas of science, thinking about how we can use plant cell walls and the biomass that they generate to replace fossil carbon. And along that way, thinking about biofuels, biomanufacturing, and how to use that. And so I think emphasizing the same point that space science can really take you new places, and you don't have to, it doesn't have to be everything you do, and it just throws up some really interesting questions and some really interesting framing for how you might think about things. And I do a lot of that work through a collection of centers, and today I'm going to talk about things mostly from Plants for Space, which is funded by the Australian Research Council. We've just completed our first year of funding and we'll be here for 7 years, and we're a large partnership of both universities, but also industry partners and government partners based in Australia, but we have a lot of collaborations globally, including Simon. And our big goals of the centre are about reimagining plant and food design to enable human deep space exploration, but with that piece of improving on Earth sustainability. And I'm really hoping today that we can talk a bit more about how those two things are very deeply connected. And so crews living in deep space must take everything with them to survive, and not just survive, but we have to enable people to really thrive and live well. And so I think of it as the ultimate bioeconomy. You have to take everything with you, or you have to create it in situ. There's very few resources you're going to have access to. And the International Space Station is what, 420 kilometers above us. While I think it's an absolutely incredible feat of engineering and of human cooperation that we have people living and working above our heads right now, 420 kilometers is actually not that far in the kind of grand scheme of things. If we wanted to have some human habitation on the lunar surface, that's 380,000 kilometers. So think about the supply chain challenges, the living challenges that come with that, in addition to the complexities of that environment. And if we really want people to go to Mars to do experiments, to spend some time there, it's around 225 million kilometers away. And even at the closest point, it can be 56 million kilometers away. What that means is that even communications become challenging. So it takes about 30 minutes of delay between having a conversation. So if people need assistance, even just providing that assistance can be very, very challenging. And the estimates are that if you were going to send a crew of 4 for a 3-year mission, which would be kind of 9 months there, 9 months back, a bit of time on the surface, you would need around 10 tons of food. And you simply can't carry that much. And even if you could kind of deliver the calories there, things like nutrition stability, like a lot of vitamins just don't have that same shelf stability. And things outside of the Earth's environment degrade a lot more quickly. So the quality of these things drop off, without thinking about things like medications and other things you might need. So it becomes kind of important to think about what we can create along the way. And plants really are a critical part of it. And if you read science fiction or you look at old NASA workshop reports, plants are recognized as being critical, both for providing that life support for food, but also for all the other things they produce, which we can often take for granted here on Earth. And these are just some images. I've taken one at the bottom is from a NASA workshop that Ray Wheeler and others organized back in 1999. So imagining how we might live on the surface of the moon, and you can see a plant growth facility kind of buried under the regolith. There's a 2005 NASA Ames picture on Mars. And again, you can see those plants protected and shielded from that really hostile environment, growing on shelves under defined lighting conditions. And those images look very much like the closed environment agriculture, particularly vertical farming, that is developing on the Earth right now. And so on the right-hand side, you can see an image from Scion Farms in the UK that's built by Vertical Future, who are also a collaborator on Plants for Space. And I think you can see how similar those are. And of course, there are differences. Obviously, the gravity is a very different thing. But there is some gravity on Moon and Mars. So we think that biology will be able to handle some of those things slightly better. Not the radiation, that is a huge problem. But if we're thinking about closed environment agriculture, Earth, Moon, Mars, there's some features we can think about. It's contained and it has a defined environment. That defined environment is really important when we're thinking about stuff that I work on and engineering plants, because thinking about how having defined phenotypes of our plants is really, really important. If any of you have ever run a field trial, you know how different your plants can look year to year, because they can be affected by weather, by pests, by gophers, whatever it's going to be. And so that beautiful phenotype you developed in a glass house doesn't always translate to the field. At commercial scale in the vertical farming, I think we get much closer to those convirons we might use to do our everyday plant science research. You get year-round production. So we're taking the seasonality out of things, and you often have reduced water usage, so you can recycle and recirculate everything. But to be really successful in this, we need new crops and cultivars designed for this environment. So a lot of the plants that are currently successfully grown in CEA are often lettuces, chilies, herbs, things like that. So if we want to move to more complete plant-based diets, and in space, it's pretty much likely we're going to be eating a vegan diet, you need things like high protein crops. You also need a lot more remote monitoring, early intervention, and automated harvest. It's very hard to actually monitor these types of farms, but you can imagine how difficult it is to even look at those upper shelves in the farm I've got on the right. But also, labor is challenging. On Earth, it's having labor for agriculture is a real challenge. In space, it's much, much harder. And so astronaut time is enormously expensive. I'm sure Simon can talk about that. And so the more we can do with remote monitoring, the more successful we'll be. But there's also some really interesting opportunities for both biomanufacturing, so using plants as a chassis for producing things, but also for using waste streams. So thinking about that circular economy idea of what we have, because all your plants are grown in one space, you have a lot of control of what's there, and so you have materials that you can use for other features, such as producing biomaterials. And so the kind of argument we would put forward is that, and this is from a review that was written as part of a UK Space Agency grant we had, we need plant science-driven crop redesign for this type of environment, whether in space or on Earth. And I'm not going to go through the details here, but it's really this idea that we can use all that amazing fundamental knowledge we have as plant scientists to think about how we can design for these particular fixed environments. And that we can sort of supercharge this process of producing these novel crops using synthetic biology. It allows us to kind of take all those years of plant breeding and domestication and say, can we really push that forward and deliver these new crops in a really timely fashion? Because we need them soon. And as I said, it's not just food, it's sort of on-demand production of materials and medicines. And this is sort of how I imagine the very futuristic version of biomanufacturing might be, is that you have an identification of need, and it might be pre-mission, as in you will need this thing, but it might be during mission, because unexpected things arise, and the further you are away from home planet Earth, the more things are likely to rise you didn't think you would need because of just chance. The longer missions, things happen. So I imagine that you would do the design, build, and test and learn on Earth. You would do all the research there, and there would be a transmission of a DNA blueprint. And so then I think about our plants, or perhaps microorganisms, as being sort of our 3D printer, where we create our new organism in situ and then produce the thing we need. So you can think about potentially, say, a medicine because someone has got sick and you're not carrying that medicine, or it's not shelf stable. So how might you do this? There's lots of different plants we might use. I have a particular favorite right now, which is duckweed, partly because it's so fast growing, and that's what you can see in this time lapse on the right-hand side here. It doubles every 2 days. It's completely edible to humans, and it's very high protein. So it's about 40% dry weight protein and has all of the essential amino acids. But because it produces clonally, there's opportunities to use it as a system for production, kind of like a photosynthetic yeast in some ways. And while people have looked a lot at using algae, cyanobacteria for producing some compounds, because this is actually a monocot that was returned to the water, we understand its biology a bit better, although it has some interesting features as well. And while I'm new to the space world, we have started to get involved with some space projects. And so this is a sounding rocket. So this is a way in which you can get some experience of microgravity to your biology without going all the way to the ISS, which is an extremely complex and competitive thing to be able to do. And so this is the Matthias 15 campaign. It's run by the German Space Agency, the DLR, and they put 20 or 30 experiments on a sounding rocket. This is flown from Esrange in northern Sweden. And you get about 8 minutes of microgravity, and then you get sample return within 24 hours. So we work with Jens Hauslager and Sebastian Fels at the DLR, who designed a system called the Mini-Weed for us, which slotted onto Matthias 15. And it's essentially 2 syringes, and one syringe had RNA later in, and one had our duckweed in. And this was replicated. And then after the hypergravity phase, RNA later was added to fix the samples, and then after the microgravity phase. So we got this very early insight into how these plants responded. And then we've done RNA-seq on that, and we're still analyzing the data, but we see already big changes to the cell wall, which kind of follows on from that work that Simon was talking about that he'd been doing, showing that you get these whole cell changes in response. So this is some kind of system we can use to do some of that early experiments. We're also very interested in how you might grow sort of larger scale amounts of food on the future space station. So this is Axiom Station, which is the commercial station that will replace the ISS when the ISS is decommissioned. And we had a recent project with the UK Space Agency and the Australian Space Agency, led by Vertical Futures, to develop a system where we could grow food for those astronauts. So it's going beyond sort of research scale plant growth to this. And they recently got to fly the test version of this on a parabolic flight, which I thought was pretty awesome. So this is them testing the nutrient delivery system. And so this gives us an opportunity to really work with engineers, work with people who are developing these systems here on Earth, and to test these for spaceflight. And finally, just a brief mention of a project that we have, which is led by Space Lab Technologies in the US, which will be an Artemis 3 deployed instrument. So Artemis 3 will return humans to the lunar surface, and we will be growing Arabidopsis, one of the Wisconsin fast plants, the Brassica rapa, and some duckweed. And the idea is they will germinate and grow on the lunar surface, and then we will add RNA later or some other fixative, and the astronauts will return the sample. So we'll have some sample return of plants that have experienced the lunar surface. There's lots of technical challenges to get us there, but it's very interesting to me that of the 3 experiments that are going to the lunar return, one is plant science. And I think that really underlines how important it's considered for the space programs. So just a quick summary. So closed environment agriculture, kind of supercharged with synthetic biology, could rapidly scale findings from the lab to the industry, and that plants are going to be a critical part of this long distance space exploration. As a cell wall biologist, I'm super excited to think about how these cell walls undergo extensive remodeling in response to the change in gravity, even though plants have only really ever seen 1G. So they've not really evolved to do this, but they have these rapid responses. And that problem solving for space is going to directly benefit us here on Earth. I think I have an awesome team, both here in Adelaide and some very patient folks over in Berkeley Lab as well, where I was before, and particularly Plants of Space for funding. And I'll just leave you with this image that came from Intuitive Machines when they were doing one of their lunar landers. They sent a photograph back. You can see Australia there, Adelaide really clearly. And I think it's pretty incredible what's going around us in the space environment right now. The advances are incredible. And with that, I'll pass over to the next speaker.
All right. So our next speaker is Dr. Veronica D'Amico, who is a full professor of environmental and applied botany at the University of Naples, Federico, where she is responsible for the plant and wood traits lab at the Department of Agricultural Sciences. So the stage is yours, Dr. Miko.
Thank you. Oh, there is a message saying that I'm not, I cannot share my screen.
Yeah, you should try now.
Okay, now it works. Thank you. Just confirm you see my presentation. Can you confirm it?
Yes, we see it.
Okay, thank you. So thank you very much for for organizing this stimulating webinar and giving me the opportunity to share my experience with you. I will focus on the role of higher plants for space exploration with a focus on radiation. And as an agronomist and botanist, my main interest is in understanding mechanisms for adaptation and with the aim of linking the structure and function, so with the ecophysiological traits too. That is one of the main goals of my lab, that is Plantae Wood Traits Lab. For space application, we are interested to see how the plants acclimate to space, because we agree that it's not only my opinion and also Professor Jenny and Simon, who spoke earlier than me, it's not only our opinion, but in general, the national agency worldwide agree that for there are critical issues that have been listed in the Global Exploration Roadmap to face for bringing the humans up to Moon and Mars, so beyond Leo. And two critical issues regard the life support and habitability, and also the crew health and performance. So we need the plants for facing these challenges, because the plants are one of the key elements in bioregenerative life support systems. So the idea is to create artificial ecosystems in which the plants, of course, are the producer. This is one of the schemes that you can find in literature and on the web. This is from the Melissa program of the European Space Agency. It's a program of more than 30 years, more or less, of which my department and university is also member. And in this Melissa program, we focus on the role of higher plants, because we've seen that they are very important for the regeneration of resources and also for the food production. One of the key issues is the choice of species and cultivar, because they depends on the mission scenario. If we are talking about a short-term mission, we can consider plants just for the integration of the crew diet. And in this case, most of the food is still brought from the earth. But if we go further and further from the earth, we need to cultivate also staple crops, because they need to furnish most of the nutritional intake in terms of protein, carbohydrates, lipids, and so on. So the plants are keys for these ecosystems, not only for the regeneration of resources and for producing the food, but also because they have an important role that is related to the psychological issues, because they are important to mitigate the stress of the mission, because they contribute to the improvement of mood, reduction of anxiety, and so on, mainly related to the green environments, the production of fresh in situ produced food, so to improve the nutritional uptake and also to improve the air quality. Anyway, so if we agree that we need to bring with us the plants, we also agree that further and further from Earth, radiation is going to become one of the main problems, and it is considered as a showstopper for the space exploration. And we need to mitigate the risk for humans, and there is common agreement for an integrated view for radio protection that includes passive shielding, active shielding, but also the strengthening of the defenses, the physiological defenses of the human body itself, with radio protectors, mitigators, but also with introduction in the diet of fresh food. The idea is to introduce in the diet of the astronauts fresh food produced on board that is richer in antioxidant and some nutritional compounds that can, of course, strengthen the physiological defenses of the astronauts. In this view, the good news is that plants are much more resistant than animals in space because of their specific structure, genetics, physiology, and so on. There are some similar aspects, but also many differences. For example, plants show high variability of responses depending on the type of radiation, the dose, the species, the cultivar within the species, but there is also common agreement that low doses that are already really high and terrible for humans can induce positive effects in plants. This phenomenon is known as hormesis, and it happens with an increased content of antioxidant, improved growth, germination, nutritional value, and so on. If we increase the dose, also the plants can start to suffer. So in my lab, we have been working for more than 15 years on radiation, and we performed many experiments with different types of radiation. X-rays is the reference radiation for space, and then we also considered EV ions on several species. And the main goals were can be grouped into three. So the understanding of radio resistance mechanism, but also to evaluate the impact on the regeneration value for the definition of crop requirements in the space greenhouses, and also to evaluate the impact on nutritional value of edible organs. This is our workflow. We established the procedure for irradiation. We have done most of the irradiation in hospitals for X-rays, but at GSI in Germany, in Darmstadt for EV ions. Then we cultivate the plants and we make the analysis in terms of growth and morphology, microscopy for the quantification of functional anatomical traits that we link with ecophysiological traits, and then we also make biochemical analysis related to the functioning of the system, especially the hydraulic system and the photosynthetic machinery, and also to evaluate the nutritional value of the produced food. I consider some of the study cases. For example, here we irradiated the tomato plants at different phenological stages with X-rays. We irradiated the seeds and then the adult plant, flowering plant, and plants with ripening fruits. And as you can see, irradiation did not stop the growth cycle up to values very, very high of radiation. But when we irradiated the adult plants or flowering plants, doses starting from 20 grays induced the early senescence, so the crop cycle was stopped. And again, when we irradiated the plants that already had the fruits, they completed the ripening. In general, we found an increase in ascorbic acid, sugars, phenolics, and so on, especially when the plants were irradiated at the ripening stage. Then we were interested to see whether the crop conditions can alter the response to radiation. And so we, in this study case, we analyzed another species and we irradiated germinated seeds because germinated seeds are much less resistant than the dry seeds. And then we incubated them at different light qualities regimes, and the light quality had an important effect on the response to radiation. For example, the red light increased the content of many compounds as soy flavonoids when radiation increased the dose. This did not happen with the other light qualities. Then in the last years, we've been involved in analyzing the effect of species, target system, different radiation sources and doses in a project funded by the Italian Space Agency. And we were here interested to see whether the low doses can improve the physiological performance of the plant. And I am not going in depth in the results, but I just want to tell you that the response was, of course, ion and dose and species specific, with carbon inducing a more tight coordination of all the traits, maybe because there was a higher sensitivity at low doses. So the plants adjusted its structure to perform better. But in any case, with the different strategies of traits coordination, at all radiation treatments, there was a good biomass production, and so no specific aberration in all the cases. So we trace we are tracing a sort of shielding strategies to apply the results also to more practical objective for shielding. And so the idea we propose is to evaluate whether there are only negative outcomes. So we have to assess the plant sensitivity to radiation and define the level of shielding based on a cost-benefit analysis, because shielding is also very expensive for space protection. And in case the plants show positive outcomes, we can assess the threshold doses for hormetic responses and then evaluate an opportunity for modular shielding approaches, maybe only in some specific phenological stages of the plant. So the idea is that if irradiation does not overcome this threshold, we go for no shielding. Otherwise, we go back in the shielding setup and evaluate again with the cost-benefit analysis whether it's worth to apply the shielding and which level of shielding. Now we are currently working with some experiments. One is based on the possibility to try to pre-acclimate the seeds with ionizing radiation before exposing them to the moon environment to see if they can adapt better to the moon environment. We have another experiment in which we are combining space radiation and chronic radiation and microgravity at the Mars simulator in Toulouse, and we are also expanding our boundaries toward mosses to use them as biofilters. Ongoing research is also considering more tightly the effect of cultivation factors. For example, we are testing in another project funded by the Italian Space Agency multi-cropping systems towards single cropping systems in hydroponics, in rice, bean, and lettuce, because the multi-cropping will be likely the solution for in space greenhouses because of a lot of technical constraints. And so, which is my take-home message for further perspective? We need to identify the threshold doses that maximize the nutritional values without too much loss of biomass. We are going to perform experiments, increase the number of experiments due to the variability of results with chronic radiation, combining the effects of several radiation types, and also using the galactic cosmic simulator, and we applied for this recently, and also we are evaluating the interaction with the cultivation factors. I invite you to visit us in Portici, in Naples. At my department, we have this laboratory for crop research for space. The responsible is my colleague, Professor Stefania De Pascale. Here, a lot of experiments or many groups of the world can use this facility that is a controlled chamber in which we have a tight control of the above ground and below ground biomass. I thank you all for the attention, and I have some pictures of my collaborators. We realized that we only have pictures together when we are eating or drinking because of celebrations. So please just consider this a small picture, and thank you for the attention.
Thank you very much to all of our speakers and moderators. And I think this time we actually got quite a diverse set of perspectives in this seminar. Now we're going to quickly move on to our Q&A session. So to our audience, if you haven't already, do submit your questions and we'll endeavor to answer them as best as we can. Okay, so first question, is astrobiology something that only NASA is interested in, or are there other space agencies such as ESA also offering opportunities for plant scientists? And this question comes from a plant genetics PhD student, and they mentioned that working in astrobiology is something that it would be an absolute dream job for them, preferably in Europe. Yes. Would any of the speakers like to share?
Yes, if you want, I can answer because I'm from Europe, so we refer mainly to the European Space Agency that has many programs, but also the other agency have programs for PhD students or postdocs. For example, I mentioned the Melissa program, who every year make calls for PhD programs in which that they are directly funded by the Melissa Foundation or through the single country agency. And as well, there are also programs for postdocs. So my advice is to just have an eye on the websites of these the different agency where you can find the call for PhD or other fellowship and so on.
Thank you. So the next question is also a general question to all of our panelists. Has there been much work studying microalgae in space, as they are typically more photosynthetically efficient than higher plants?
Yep, there has been microalgal work. A lot of the most recent research is with plants. One of the goals in the background of everyone's research is we would like to all be contributing to the bioregenerative life support systems. So a lot of people's research, even if it's basic research, is to understand what's happening with plants with the goal of putting them into that sort of the food, oxygen, nutrients loop. And so one of the criteria, which is very easy to sort of, as a practicing plant scientist to forget, is you've got to grow things that the astronauts would like to eat. And algae don't tend to be high up on their lists.
Thank you. Yeah, thank you, Dr. Gilroy. Next question is also directed to Dr. Gilroy. Is it possible to use a magnetic field or a magnetic field gradient in space to substitute for gravity in order to suppress microgravity? And have you actually tried that?
Oh, it's an excellent question. So we've not tried it. It is possible to replace some of the force of gravity with a magnetic force. There are these things called magnetic levitators, where if you go onto like YouTube, you can see pictures of like frogs floating in midair on a magnetic field. So it is possible to have that kind of force from a magnetic field. The magnetic fields are enormously steep, and it's just not realistic to generate that kind of thing at a level where it would be sort of a more than a research tool. So yep, you can make gravitropic-like responses, magnetotropism, but the gradients of the magnetic force are fantastically high.
Excellent. So next question is a general question to all of our panelists. What differences occurs in plant growth speed in space as compared to on Earth with respect to growth hormone effectiveness? I'm assuming they're referring to treatments of growth hormones on plants.
This is a Simon question.
I think I can take this one again. So a lot of the experiments which you assume must have been done for spaceflight have not been done. We're very much in the very beginning stages of the exploration of what happens in space. And some of the experiments which you imagine should be really, really trivial to do are very, very complicated to do in space. So thing like spraying a plant with auxin, the spraying bit of it is fantastically difficult to do in space. Just you can't squirt stuff around, aerosols float around forever, all of that kind of stuff. So hormonal treatments have not really been explored very heavily in space, but at the level of growth, of course, there's lots of data now about plants growing in space. A lot of times the plants are growing more slowly in space than they are on the ground. The question then is that something intrinsic with space, or is that just that those plants are not happy and we're not good space gardeners? And if we knew how to set up the growth chambers in a much more sophisticated way, maybe plants would grow faster in space. And there are some examples where plants do grow faster in space. So, but a lot of the background of space biology at the moment is stress biology. And one of the big questions is how much of that is us as how we're growing the plants versus what plants really fundamentally experience in space.
Yeah, and just to add to that, so they've shown that I think some microorganisms grow faster because without the microgravity, the cells just expand faster when they're growing. And so there's these questions about if that would apply to plants too, if we could grow them in sort of optimal conditions. And that comes back to that other thing that Simon mentioned with how much of what we can recreate here on Earth. If we could obviously do the experiments here, then life would be much more straightforward because we would be able to do a lot more replicates. We would have a lot more control. We would have, professional plant scientists running the experiments rather than having these uber complex systems where we hand off experiments very far in advance and we rely on these amazing astronauts who are doing a million other things to do these experiments. I think it's really interesting to try and unpick what we can learn here on Earth and what we really can't, and therefore we would have to develop new systems for doing in space.
Yeah, if I can also add something, there is a sort of paradox in plant growth in space, is that at the early stages of, let's say, cell proliferation and so on, there are some disruption of mechanisms, but then they are overcome and the plants grow well as well. If they are able to overcome these very early stages, then it grows. Also because I remember when in my first experiment, when I was a PhD student on the Photon M2, that is a satellite that makes several rounds around the Earth and then comes back, we germinated the seeds and we looked at the cell wall organization, and the microfibers of cellulose were completely scattered, so they lost their organization. And this created the loosening of the cell wall that, as Professor Mortimer just said, created the less mechanical resistance of the cell wall, so they grew larger. But then there were other factors interacting. In that specific case, there was a drop in the temperature because the satellite had not temperature control tight, but it was a passive control due to the functioning of other experiments. And one of the main experiments suddenly stopped, and so the temperature dropped down, and so our process of growth was blocked. So as you know, it's very difficult to make experiments in the space, and you have just one shot, and you never know if you can have a reflight. In that case, we did not have the reflight, and so these big, let's say, question marks remained unanswered for us.
Thank you very much. Unfortunately, with that, we'll have to conclude our Q&A session and our webinar for today. Again, thank you very much to all of our speakers, my fellow moderators, and to our audience. And if you have any pressing questions, I would highly recommend that you interact with our speakers through their provided social media platforms. And please stay tuned to our next webinar. Thank you.
Yes, thank you everyone again for joining today. And again, a very special thank you to our speakers, moderators, and to all of you who attended today's webinar. You can watch this full webinar recording on our Plantae YouTube channel. And I hope that you have the great rest of your week. And we hope to see you on the next Plantae webinar. Bye, everyone.